Hello, in this video, we're going to continue talking about the adaptive immune system by talking about how B cells and T cells get activated. So in a previous video, I briefly mentioned how B cells govern humoral immunity and how T cells govern cell-mediated immunity in the adaptive immune system. But I didn't mention that B cells and T cells have to be activated for them to perform their functions. And so in this video, we're going to talk about how B cells and T cells get activated. So B cells and T cells have receptors on the surface of their cell. And so here is a zoom in of the cell membrane of a B cell. So imagine we are zooming in right on the surface of a B cell, and right here we're zooming in on the surface of a T cell. They have these receptors, and these receptors allow them to recognize specific antigens. Remember, they're part of the adaptive immune system, so we're going to be looking at specific antigens. And when a B cell slash T cell recognizes an antigen, that is the first step in activation. So B cell receptors, they look like this. They have this Y shape similar to an antibody. Basically, they are an antibody. And they're just attached to the surface. So they just look like that. T cell receptors are just two straight lines like this. So they have kind of like an LL shape, I guess. And again, same thing, they are found on the surface of the T cell. Now, there are two types of T cell receptors. We call them the CD4 and the CD8 T cell receptors, which will be important when we talk about actual activation. So just keep that in mind. Now, for B cells, there's only one type of B cell receptor, and that's this guy. So let's start with T cells. T cells are categorized based on their receptor, so we call them the CD4 T cells or the CD8 T cells. So if you are a T cell with a CD4 receptor, you're called a CD4 T cell. And to activate its CD4 T cell, it needs to interact with an antigen. Remember that these receptors recognize antigens, and this antigen has to be presented on a different receptor called an MHC class II receptor. And MHC class II is found on only certain white blood cells. So things like your macrophages, dendritic cells, and B cells, they're the ones that have these MHC2 receptors. So here is a dendritic cell, a special type of white blood cell. It is going to be presenting an antigen on its MHC class II receptor. So here's the MHC class II receptor, and here is the antigen. Now, when I say presented, it literally means that it is showing the T cell this antigen by basically holding it in the MHC class II. It's kind of like a little hand. It's giving it to the T cell. And the T cell is going to recognize it using its T cell receptor CD4, okay? And when this interaction happens, the T cell becomes activated. And when a T cell becomes activated, if it is a CD4 T cell, it is going to become a bunch of different types of helper T cells. So we call them TH, H for helper. There's a bunch of different types, and then there's also the memory helper T cells. And these are going to be important for remembering the bacteria or virus for later. Anyway, these helper T cells, all of them, their main job is to be the cheerleaders, and they're going to be activating, sorry, that should say activating, not active, activating other white blood cells that are found in the body. Now, what about the CD8 T cells? So CD8 T cells are T cells that have the CD8 receptor. And what they're going to be interacting with is an MHC class II receptor. MHC class II, by the way, is found on all of your cells. So my liver cells and my lung cells and all of my white blood cells, every cell in my body, with the exception of red blood cells, every single cell in my body has MHC class I. So here is a cell, any cell. It is infected. What it's going to do is show the antigen in its MHC class I. It's going to present it, and it's going to present it to the CD8 T cell. So the T cell becomes activated through this interaction, and this CD8 T cell becomes what we call a cytotoxic T cell. Cytotoxic T cells are very important because they will basically kill any infected cells that are in the body. And of course, they're going to identify those infected cells by looking for that specific antigen. By the way, when a T cell gets activated by an antigen, it will only kill things that have that antigen. So again, very specific. And how do these CD8 T cells or cytotoxic T cells kill another cell? they will release these granzymes and perforins. These are enzymes that basically degrade the cell that is supposed to be killed, and it will cause that cell to go through apoptosis, which is cell suicide. Now let's go over the B cells and how they're activated. So B cells are activated two ways. One is without the help of a T cell. So a B cell can interact with an antigen using its B cell receptor, and the antigen can be on the actual bacteria or virus or just floating around, doesn't really matter. But basically the B cell gets activated and starts to produce these antibodies. And that's it. The antibodies that are produced, by the way, are called IgM antibodies. And that is really important because IgM antibodies are very fast in their production, but they're not the most effective. So just a quick, I'm just showing you this real quick. So when you are infected with something, your body will always produce IgM first, and that is the primary antibody that is produced because it's very quick, but it also is not as effective as IgG, which is the kind of main antibody that our immune system relies on. And so your immune system will make IgG later. Now notice that when you are infected for the first time, you make some IgM and then you make a bunch of IgG, and that's it. Now, remember I said your immune system is capable of remembering later on. And so if you were to be infected a second time, your body is able to make tons of IgG first instead of IgM. And that is going to be significant because now the likelihood of you getting sick or having any symptoms decreases because you basically produce tons of antibodies, IgG antibodies, from the beginning. Anyway, how do we make those IgG antibodies? That is through B cell activation with the help of a T cell. So going back here, this is B cell activation without a T cell. This is B cell activation with a T cell. So a B cell is floating around and it's going to meet a T cell. Okay, that is a helper T cell. So what the B cell does is it is presenting an antigen on its MHC2 receptor. B cells have the MHC2 receptor. And so the B cell is holding the antigen right there, and it's going to present it to none other than the helper T cell. And so the helper T cell, remember, has CD4, so MHC2 and CD4 are compatible, and so they're talking to each other. Basically, the B cell says, hey, I found this. Is this significant? And the helper T cell says, yes, that's what I've been looking for this whole time. And so what the T cell does is it releases cytokines, and that activates the B cell. And it causes the B cell to go through what we call clonal expansion, meaning the B cell will replicate multiple times, several times, and it will make clones of itself. Some of those clones become memory B cells, which are useful for later, to remember the pathogen for later. And then the other B cells are going to be converted into plasma cells. These plasma cells are factories that produce tons and tons of IgG antibodies. And that is how we get that second peak of IgG. And again, because we have memory B cells that are produced when you get infected a second time, boom, you make tons of IgG because you have all of these memory B cells ready to go. So that is pretty much how B cells and T cells are activated and their whole role in the immune system. Thanks for watching, and I'll see you in the next video.